It's easy to take soil for granted. We walk on it, plant in it, and hardly pause to wonder what's going on beneath the surface. But soil quality is actually a massive deal, especially when you think about the challenge of feeding nearly 10 billion people by 2050 without wrecking the planet.
That's what blows my mind. Most folks don't realize soil isn't just dirt. It's alive. It's got this whole web of chemistry, physics, and biology happening at once. When we talk about soil quality, we're really talking about the soil's ability to support plants, store carbon, and keep our ecosystems healthy.
Exactly. And it gets tricky because not all soils are created equal. And even what counts as good soil can change depending on the crop or the environment. There's a ton of debate about how to even define soil quality.
Totally. For instance, the ideal pH for one crop might spell disaster for another. So, any attempt to measure soil quality needs to be flexible, but still rooted in real, tested indicators. Which brings us to why scientists develop these soil quality indices in the first place.
So, let's get into the nitty-gritty. What are those indicators scientists keep talking about? What do they actually measure to figure out if a soil is healthy?
They break it down into chemical, physical, and biological indicators. Chemically, you're looking at things like pH, organic matter, nutrients basically. Is there enough food in the pantry for plants?
And then physically, you've got texture, bulk density, aggregate, stability. Those tell us if roots can grow, if water drains well, or if the soil's going to erode in the next big rainstorm. Kind of like checking if your house is built on solid ground or a swamp.
Exactly. Then biologically, scientists look for signs of life, like enzyme activity or microbial respiration. If the soil's teeming with microbes and enzymes, chances are it's a more resilient and productive system.
It's wild how interconnected all those are. You tweak one, and the others often shift too. Speaking of which, I'm curious, how do they combine all these moving parts into a single score? So with all those indicators, chemical, physical, biological, how do researchers actually pull them together into something meaningful? I saw there are different types of soil quality indices, or SQIs.
That's right, and it can get pretty technical. There are additive indices, where you just average all the standardized parameters. Then there are weighted indices, like SQIW1 and SQIW2, which give more importance to certain groups, say, nutrients versus physical structure.
And I noticed some methods use principal component analysis, almost like letting the data itself figure out which indicators matter most. That's a smart way to deal with all the possible overlaps and correlations.
Totally. Principal component methods basically distill the mess down to the strongest signals. What's neat is, depending on which index you use, you might get a different answer about which soil is better. It really depends on what you care most about: nutrient availability, physical resilience, or biology.
That actually reminds me, let's talk about the two soils from Puerto Rico that were at the heart of this particular study. Their story is a great example of how these indices play out in the real world. So this study focused on two Molisols soils in southern Puerto Rico, the Yauco and San Anton series. From what I gathered, Molisols are generally rock stars in the soil world, super fertile, high organic matter, great for agriculture.
Yeah, and even between these two, they've got different personalities. The Yauco series is loaded with calcium carbonate, making it pretty alkaline and rich in organic matter. San Anton, meanwhile, has more clay and tends to pack in more micronutrients, like copper and iron.
What's really interesting is how management, like tilling or leaving the soil alone, affected them. The researchers compared conventional disc plowing with a no-till approach, taking samples from shallow and deeper layers.
I loved that they looked at everything: chemical tests, physical structure, even the mineral makeup, using fancy tools like FDIR and XRD. Turns out, deeper soil layers almost always had lower quality scores, but tilling didn't make as big a difference as you might expect.
Funny how sometimes what you think will matter most, like tillage, ends up being less important than depth or even the soil's parent material. So, how did these soils actually score? Here's where it gets cool. Depending on which index you use, you get a different winner. The additive index, which averages everything, gave San Anton the edge for overall quality, mostly because it scored high on nutrients and micronutrients.
But then, when you look at indices that put more weight on physical parameters, like bulk density and aggregate stability, Yauco comes out on top. Its structure and organic content shined there, even though its high pH lowered some micronutrient scores.
Also, I found it fascinating that both soils lost quality as you sampled deeper down to 20 centimeters. Makes you think about how most root activity, organic matter, and biological life is concentrated near the surface.
Exactly. And honestly, the fact that tillage didn't dramatically lower soil quality scores in the short term is a reminder that soil responses can be slow or subtle. It's not always a clear-cut story.
So, wrapping up, it feels like the best approach is a mix. Choose your indicators and indices based on your specific goals, whether that's high yields, resilience, or environmental health. And always, always keep an eye on what's happening beyond the top few inches.